morning, children. This is Aniruddha Mount Literacy School. In the previous session, we discussed about mitosis, and today we are going to talk about meiosis. It is a kind of division of diploid germ cells that reduces the chromosome number by half, forming haploid daughter cells. In mitosis, as we have already discussed in previous lecture, that in mitosis is also known as equational division. Why? Because the number of chromosome remains the same in the daughter cells also. But in meiosis, the number of chromosomes become half in the daughter cells. In mitosis, only 2 daughter cells are formed, but in meiosis, 4 daughter cells are formed, and they all have half number of chromosomes. So, meiosis process takes place only in gametogenesis process, that means the formation of gametes, and it leads to the formation of haploid cells that are known as gametes, male and female gametes, and this is found only in sexually reproducing organisms. And ultimately the gametes combine together to form again a diploid structure. This process is known as fertilization. The key features of meiosis: it involves 2 cyclic processes, meiosis 1 and 2nd, but only a single process has DNA replication. That means DNA duplicates only in one process. Now it involves the pairing of homologous chromosomes and recombination between them. That means the homologous chromosomes which lie together parallel to each other, they combine together, they cross over together, and they recombine their chromosomes again. This is known as recombination. Meiosis 1 begins after the replication of the DNA, the entire chromosome, to form identical sister chromatids at the S phase, as we have already discussed about the G1 phase, S phase and G2 phase. And S phase is the phase in which DNA duplicates to form 2 copies of itself. 4 haploid cells are formed at the end of the meiosis 2nd. Stages of meiosis. The stages of meiosis are divided into 2 sections, meiosis 1 and meiosis 2nd. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2nd has prophase 2, metaphase 2, anaphase 2, and telophase 2. And the diagram here on the structure you can see. The prophase again is divided into 5 sections. Those 5 sections are known as leptotene, zygotene, pachytene, diplotene, and dikinesis. And in these segments, different segments, you can easily see different kind of changes in one organism. So in leptotene, the nuclear envelope becomes starts decreasing, it starts degenerating, and ultimately the duplication process of chromosome also starts and ends in leptotene stage. In the zygotene stage, here we can see the chromosomes, 2 chromosomes which are being formed, they come together and become parallel to each other, and they start making a synapsis. That means the combination of both the chromosomes. Then the pachytene stage, they lie together, and one hand of one chromosome, that means sister chromatids, crosses over with another chromosome. Synapsis completes here, and crossing over process starts in pachytene. The diplotene stage, the chiasmata, that means the X-like structure of 2 sister chromatids is still visible. Synaptonemal complex starts disappearing and chiasmata becomes visible, and ultimately in dikinesis the chiasmata is degenerated. That means both the sister chromatids separate from each other and their chromosomal material exchanges. And this was the prophase 1. So here you can see one by one all the stages. The chromosome becomes more condensed in zygotene stage, and similar chromosome starts together as a synapsis with the help of a complex structure called synaptonemal complex. The pachytene stage, here you can see that the comparatively longer phase, it is the bivalent chromosomes is split into similar chromatids. This stage is called as tetrad. And the crossing over process occurs in this pachytene stage. The next stage is Diplotine stage, and the Diplotine stage dissolution of synaptonymal complex occurs. The combined homologous chromosomes of the bivalent separate from each other, one except at the site of the crossover. That X-shaped structure is still visible, and that is known as chiasmata. In the dikinesis process, the chiasmata is completely terminated and chromosomes are fully condensed, and the spindle formation starts, and the nuclear envelope disappears. Now we come to the metaphase, that is the second stage of meiosis 1, metaphase 1. Spindle formation is completed in this stage, and both the chromosomes come at the equatorial plate. Anaphase, when the chromosomes start separating from each other, just like mitosis. And the telophase, when both the chromosomes separate from each other, and from there the cytokinosis also occurs and their cytoplasm is also separated and two daughter cells are formed. That is known as telophase 1. Then the process comes meiosis second, which just looks like mitosis, and it has also 4 stages: mitosis second, prophase second, metaphase second, anaphase second, and telophase second. And here you can see in this particular stage that the prophase second, it is initiated immediately after the cytokinesis. The chromosomes again become compact, nucleolus and nuclear membrane disappear in both the nuclei. In this stage, DNA does not replicate, but the DNA chromosomal number or the DNA number becomes a half. The chromosome align at the equator and microtubules from opposite poles of the spindle get attached to the kinder cores of sister chromatids of the chromosomes. This is metaphase second. Both the structures you can see, prophase second and metaphase second, all together. Then comes the anaphase, where metaphase and anaphase both can be seen. It begins with the simultaneous splitting of the centromere of each chromosome, which was holding sister chromatids together. Thus, they move towards opposite poles and the chromosomes are separated. The metaphase second and anaphase second both can be seen here. The telophase, that means the last stage of meiosis second, the telophase second, where both the chromosomes completely disassociate from each other and they are completely moved towards two poles. The two groups of chromosomes once again get enclosed by a nuclear membrane envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells, and they are known as 4 gamete cells. If this process takes place in female, then it is known as female gamete. If this process takes place in male, then it is known as male gamete. And they all have number of chromosomes half. Overall steps of meiosis 1 and meiosis second you can see here on your screen. By pausing the screen, you can easily see and understand both the processes. Now the significance of meiosis, as I have already discussed about this before starting the lecture. It conserves the chromosome number of each species. It causes the genetic variation because it is the process in which the crossing over occurs, in which the chromosomes are the chromosome material is exchanged from each other, and that is why it causes the variation in the population of the organism. It is important for the evolution because you know that the variation is very important. Sexual reproduction is a better mode of reproduction because it causes variation, and that variation is the basis of evolution. The differences between mitosis and meiosis, here you can see we have already discussed so many times about the mitosis and meiosis, so the differences are there on your screen. You can pause the screen and read the different differences between mitosis and meiosis. And here this particular chapter of cell division and cell cycle ends. Thank you very much and have a nice day.